Hello everyone, my name is Dr. Bruno Andrade and I'll talk about my poster session BioPepTool, a high throughput screening tool for discovering peptide bioactivity. So, as you well know, SARS-CoV-2 is the virus causative of COVID-19. For this, we can see that this viral genome is composed by a 29.9 kilobase in size and present 82% sequence identity with the first SARS-CoV. In addition, 11 protein-coding genes and 12 expressed proteins. In this work, we focus on spike protein, which is responsible for interacting with the human ACE2 receptor and allows the virus entering the cell. This spike protein presents a RBD region, which is the main responsible for do the binding mechanism with the ACE2 receptor. Using this RBD region and other regions in the spike protein, we can work for developing drugs against COVID-19 and antibodies and peptides for viral prophylaxis and vaccination. So in our COVID-19 research, we basically use peptide databases for do peptide modeling by using AI. So use this peptide database of antimicrobial activity with this peptide with no activity against bacteria and virus. We use this peptide against the RBD region of the spike protein of the SARS-CoV-2 virus. So the main aim of this work is for viral prophylaxis. So we focus basically on the RBD region for blocking the RBD AC2 interaction. But this can be used too for furin cleavage site peptides as well as TMP or SS2 site peptides. And then we can use this peptide for developing products for surface prophylaxis. And we can do medicine prophylaxis use these peptides too. Basically, our peptide discovery pipeline is composed by a peptide database with now sequence of peptides from GenBank and CBI GenBank and other from papers available in many databases. So we got this peptide sequence and do a 3D modeling using modeler software. And after we do docking against the RBD region of spike RBD using HPEPDock. After this, we basically collect the docking energies and peptide features for developing a training database to do a surrogate model. With this, we start a new population of peptides and sorting a method for sorting a sequence of peptides. And after we propose this new sequence peptides against the same receptor in the same region of RBD spike, and after we compare if the new peptides present a best in docking energy in comparison to the training database. If it's okay, we got the result and we can propose the new peptide for in vitro and other bench experiments. If not, it can restart the method and do another sequence of peptides. So basically we use this AI prediction with now peptides and docking with HPEPDock server, and after we have this for RBD region and doing new peptides, surrogate peptides for blocking RBD region of the SARS-CoV-2. So we have peptide candidates with best hits, which is comprising for a peptide with more negative energies in the docking results. So we propose this peptide for surrogate assisted models for do new peptides for blocking this RBD region of spike protein of the SARS-CoV-2. So as you have in our results, we have this first peptide, 411. This is a natural peptide, which we have a 230 minus 230 kilocal mole in the RBD region of the spike protein. So we have this interaction map here. And our best surrogate model is peptide 230. We have a better energy value of minus 254 kilocal by mole and per mole in the region of RBD in the semi-protein. So we have this map in the semi-region and with best energies. So as conclusions, our surrogate model is able to predict peptides with great potential. Peptides with higher RBD affinity can be directed docking validation and in vitro and in vivo experiments. And our tool can be easily applied for other targets, for other viruses and microorganisms, as well as our peptide BioPepTool is available on the GitHub. So in the next step, we hope to integrate an AI peptide modeling with AlphaFold 2, build a 90 microbial structure peptide database for viruses, bacteria, and fungi, and replacing the HPapeDoc for our own peptide docking algorithm, and provide a web-based interface freely available for all users. So this is our team composed by researchers and students, and this is our funding agencies. And we'd like to thank you for your presence and attention, and I hope you follow us in our social media. Thank you.